So after we talk about the challenges, I'll just go in with the hypothesis and say, so with this research, they're really looking to see if patient-derived organoids would serve as a good model for Alzheimer's disease. And if they did, could we then treat it with beta and gamma secretase inhibitors to then decrease amyloid and tau pathologies that show up within this disease? The techniques that were used in this research included the maintenance of induced pluripotent stem cells. These were created from human fibroblasts, and one of them was caused by duplication in the gene amyloid precursor protein, and another was generated using Sendai virus to overexpress certain genes in fibroblasts. Either way, these iPSCs were cultured on mouse embryonic fibroblasts in a media that promoted their growth. They were then differentiated mechanically under a microscope, and after these iPSCs were dissociated into a single-cell suspension, the mouse embryonic fibroblasts and the iPSCs were separated on a gel-coated dish in which the embryonic fibroblasts would attach and the iPSCs wouldn't, and so then they would be collected. From here, the researchers needed to create neural organoids, which are sometimes referred to as 3D cultures. And so first, embryoid bodies were first formed by loading 12,000 iPSCs per of the 96 cone-shaped well plates that were pre-coated with plurionic acid. They were then maintained in a media that promoted neuronal lineage, and for the first 3 days included a BMP signal inhibitor. These organoids were then transported to petri dishes so that it would prevent the fusion of the separate organoids, and then it was cultured in a medium that was designed to promote neuroepithelial formation. And then from this point on, it was kept in a media that would promote the further growth of the organoid. From here, the organoid tissues were processed for staining and imaging, and so at day 60 and 90, they were fixed in paraformaldehyde and cryptoprotected. From here, an immunolabeling process called immunohistochemistry took place. Here, primary antibodies detect proteins within a tissue section, and secondary antibodies are used with a signal molecule for signal amplification. Here, a detergent is used to permeabilize the sections by lysing the cells and solubilizing membrane proteins. And to prevent nonspecific binding of antibodies, blocking is performed with a horse serum solution. And this was followed by the incubation of these samples with the primary antibodies. The proteins that were looking to be detected were MAP2, which is microtubule-associated protein, which is a neuronal protein marker, 4G8, which labels amyloid beta, as well as amyloid beta itself, and phosphorylated tau, which are both markers of Alzheimer's disease-like pathologies. Early endosome antigen 1, which marks endosome abnormalities are common phenotype of Alzheimer's disease, as well as Cleves caspase 3, which is an indicator of apoptotic cells. The sections were then incubated in secondary antibodies, which were prepped in horse serum, how do I say this? I don't know how to say it. Nuclear immunoreactivity, as well as conjugated to cyanide dyes. They were then imaged with laser-scanned conofocal microscopes by researchers that were blind to the experimental conditions, using both the control and the familial Alzheimer's disease organoid sections in the same batch. Furthermore, thioflavin S staining is done, which is used to detect any form of amyloid formation. So to do this, sections were incubated in potassium permanganate dye. They were washed, then destained in potassium metabisulfate and oxalic acid. Then in the dark, the tissue sections were incubated in thioflavin S solution and developed with ethanol. The slides were then cover-slipped, and these fluorescent thioflavin S signals were imaged with a laser scan conofocal microscope as well by researchers blind to the experimental conditions.